Jane Chu. I'm an infectious diseases physician at Duke University. I also do research in infective endocarditis, which is usually a bacterial infection of heart valves. And I also work on educational initiatives, mostly with Amanda, who's my partner in crime at Duke. We work on career development programs for faculty members, fellows, medical students, and even undergrads and high school students, as we talked about today. So my caveat to the presentation today is that I'm not a virologist, I'm not an Ebola expert. So I'll present Ebola from the perspective of an infectious diseases physician. And if you have questions along the way, please ask. So what is Ebola? Ebola is a virus, and it belongs to the family Filoviridae, or filovirus. And the term philo comes from the Latin word phylum, meaning thread-like. And this is apparent in this micrograph here of Ebola virus, where you can see that the virus has a very thin thread-like structure. Of course, what you can't see here is the outer envelope of the virus, which is the outer portion that is studded with glycoproteins. And these are the glycoproteins that are used to attach to the host cells. And then the inner portion of the virus has the capsid that contains all the nuclear material. So this is a single-stranded RNA virus, and there are actually 5 species: the Zaire, Sudan, Ivory Coast, Bundibugyo, and the resting agent species. It's the Zaire species that is causing the current outbreak in West Africa. And as you can probably imagine, the first 4 species here are species that are found in Africa. And actually the 5th one, the resting agent, originated and is only in the Philippines. Question? Yes.
So could you say that the different species is a similarity with the different strands? So it's different strands of Ebola? Is that the same?
So if you were to do a molecular analysis, you would be able to distinguish these different species, and they can go in and say, yes, this particular outbreak in wherever, whether it be Sierra Leone right now, that would be the Zaire. Versus you could go somewhere else or go to a different point in time and identify other species.
Just slightly different RNA sequence.
Correct.
Wasn't there a Marburg?
Yes, that's a great point. Yeah. So the Marburg virus is also a filovirus, and they're both classified as hemorrhagic viruses.
Can I just ask kind of a related question here? Is it even appropriate can we say viruses are a species? I mean, if we're looking at phylogeny, I mean, do they fit in any one of our categories of living organisms, and therefore, have all of the classifications down to species?
So I guess maybe is this a question about the use of the term species and classifying these 3 different types of Ebola virus?
Yeah, just in general, is it? Is that how it's normally referred to as a species virus, and then and as such, does it fit in the phylogenetic tree somewhere?
So they're based, these different, let me just call them types for the moment. These different types of the Ebola virus are based on the RNA sequence, so molecular analysis. Is it correct to use the taxonomy of species? I don't know. Yes, certainly you can divide bacteria into different families, et cetera, species, genus, species. Does the same apply to viruses? I'm not sure. I think that certainly the taxonomy in bacteriology is confusing and changing all the time, particularly with the ability to sequence these organisms. So I don't know. Great question. Maybe we should just call them 5 different types for now. Any other questions? Any other input from this gentleman? No. Okay. So these filoviruses were first discovered in 1967 after an unfortunate outbreak in Germany and Yugoslavia, where they had imported monkeys that were infected with the virus to to a vaccine factory, and there was a huge outbreak of illness among these
It started out as a simian virus.
simian in primates.
And it switched to immune species.
So, isn't it a simian is already affects us?
Yeah.
So it's not really a simian, I mean, it should be similar to types.
So, so certainly primates, non-human primates can get infected by this virus, just like humans can. So the Zaire species was discovered in 1976 and has caused multiple outbreaks since then. And the current outbreak is the largest outbreak to date. I believe the second largest was maybe 600 cases of infection. Currently, as of last week, 14,000 cases, 5,000 deaths. And of course, this is, as you can imagine, an underestimate because these are the ones that we can actually count where patients are being managed in a hospital or semi-hospital setting.
What is the reason that it's so many more cases? Do you know?
I think it's a complicated question related to epidemiology. So the natural history is that these outbreaks occur and they have been occurring, and then they die out as the transmission process somehow just peters out.
I think a lot of them, it kills them so quickly, they don't have time to spread.
Well, or just maybe the global travel or something.
They said that highways were the biggest problem as far as people getting sick and they can transport them
Well,
to
and I
other
heard
cities.
they didn't like that it was Ebola, so they did.
Well, it's-
Yeah, it mimics cholera. I think the strain mimics cholera at first, right?
I thought it was malaria.
Well, certainly with the way that the world has changed in terms of people being able to jump on airplanes, there are no diseases that are specific to any one area anymore. Has certainly changed the epidemiology. So the current epidemic is occurring in West Africa, predominantly in Sierra Leone and Liberia, and also in Guinea. It has been difficult to really do a good count of the number of cases. And so reservoirs. So what are reservoirs of infection? These are either animals or things that can harbor the infection but don't actually get affected by it. So for a while it was thought that non-human primates such as apes and monkeys were reservoirs, but they're not, because in fact they do get infected and they die just as easily as humans do because of this infection. And the greatest epidemiologic link to reservoirs has been made with bats. So there has been, they've done some gene sequencing of viruses in bats and found that they're able to identify an Ebola virus in a bat, and also there have been epidemiologic links between bats, bat hunting, that kind of activity, and outbreaks in Ebola. How is this infection transmitted? It's transmitted through direct contact with blood, body fluids, and the skin of patients infected with Ebola. It's believed that the most infectious body fluids are blood, feces, and vomit. And fomites are also a potential source of infection. So basically the virus sitting on surfaces, contaminated surfaces. And it's believed that the virus can survive for probably a couple of hours on a dry surface and on over a couple of days in liquid materials. And we don't believe that there is any respiratory or airborne transmission of this virus, unlike, say, tuberculosis. And so the pathogenesis of this infection is similar to the pathogenesis of a lot of other viral and bacterial infections, in that it starts out with cell entry. So the virus enters through either the mucous membranes or through breaks in the skin, or, in terrible cases, direct injection in nosocomial cases. But the cell enters into the body, causes some cell destruction. It likes the cells of the immune system, so the macrophages, the monocytes, the dendritic cells, and these cells then home into the lymphoid system, go to the lymph nodes, and also to the lymphoid organs like the spleen and the liver, and then they multiply there and then disseminate out to the rest of the body. This infection activates the inflammatory response, and what people suffer from is a systemic inflammatory response, ulcers in the medical world, or sepsis. And what happens is that because of this infection, the body responds by releasing cytokines and chemokines. And what these are these small particles that cause a variety of things that are related to the inflammatory response, such as fever, but also importantly, leaky blood vessels. So what happens is that our blood vessels normally have these tight junctions between the cells so that only a few substances are let in and out. But what happens in sepsis is that these junctions become loose, and then the fluid that normally stays inside of the intravascular space escapes out. So what happens then when the fluid escapes out, people drop their blood pressure. When people drop their blood pressure, the organs don't get perfused, they get end organ damage, hypotension and organ damage. And that's really one of the big players with this disease.
What type of cell does the Ebola target?
So it's really so the Ebola virus targets these immune cells, and it goes in, hijacks the machinery, produces its own cells, and then it escapes. And what kills people is this systemic inflammatory response, in that people die because they become hypotensive, meaning they drop their blood pressure. They have an organ failure because they're not perfusing their vital organs. In some severe cases, they have coagulation defects. So part of the inflammatory response is activating the coagulation cascade. And so people can have frank hemorrhage. Now, it's called a hemorrhagic virus, which is a little bit unfortunate because it's really a minority of the patients that have frank hemorrhage. And in most cases, there is evidence of bleeding, and it's seen as some rectal bleeding. So it's only patients who have this really terrible case of Ebola that will have the frank hemorrhage.
Should be more like the HIV virus in that it affects the immune system rather than the blood vessels.
Absolutely. So it enters very similar to the HIV virus. It goes and gets disseminated to the lymphoid organs, very much like the HIV virus. And even like a lot of bacterial infections, death is not necessarily because the bacteria or the virus itself destroys organs, cells, whatever. It's really the body's inflammatory response. So how does it wreak its destruction? Through the systemic inflammatory response, hypotension and organ damage, coagulation defects with frank hemorrhage in severe cases. And then the virus has ways to impair the adaptive immune system. So with a lot of infections, if we really rely on our own immune system, despite all the antibiotics and antivirals that we have out there for infection, we really depend a lot on the human immune system to clear infection. So activation of the B cells, the T cells, and the antibodies. And people who are not able to develop these antibodies to the Ebola virus will die of it.
So, for instance, the main thing causing damage is a systematic inflammation response. Is it treated with anti-inflammatories like corticosteroids and so on?
So those are not standard treatments. The standard treatments are really sustaining, what has been termed out there supportive care, which sounds really lame and wimpy, but it's really important to be able to maintain the intravascular blood volume to help the patient just get through that